Good morning, everyone, and welcome back for our second day. Did you all have a good day yesterday? So for folks who are new and don't know me, I'm Jen Colby. I'm the Executive Director of the Northeast Pasture Consortium, and we've been running research presentations and research discussions and all kinds of great things. And we're just carrying that through today with our first speaker this morning, Dr. Stephan von Vliet from Utah State University. He spoke 2 years ago for us online, and we had a great group of folks who joined us, and they wanted more. And we knew that Stephan's work is so relevant to the pasture-based work that we're all doing. And I hear questions, and he's got answers. Not that you have answers. Nobody has answers, but you're asking the questions too. We needed folks to ask the questions that Stephan is asking. So we asked him back, and I hope you enjoy Dr. Von Vliet. Thanks.
Thank you so much. Thank you so much. Good morning. And I'm happy to be here on a rainy day. It reminds me of back home. I'm originally Dutch, so I'm from the Netherlands. So this is about the weather 200 to 300 days a year. That's right now I'm in Utah, so a little bit drier. But so I'll talk to you today about our work that we've been doing on how different grazing and finishing practices impact the nutritional quality of meat, with a focus specifically on fatty acids as well as antioxidants that make their way from the plant into the meat. So if we look at my lab's research interest, we're really interested in this central question: do more sustainable production practices also improve animal health and nutrient density of the foods that we consume? To that end, we often work together with soil scientists and ecologists and animal scientists. My background is originally in muscle biology. I'm studying the muscle of animals now, but also have a background in human health. So oftentimes we take these foods and feed it to people too in randomized controlled trials. And really, if we zoom out to the bigger picture of it, is that there's this particular interest notion in regenerative agriculture, agroecology, as we typically call it in science, the wedding of agriculture and ecology, that there's a relationship between soil health, plant health, animal health, and human health. And while this intuitively makes sense, and probably many farmers would observe, there's actually not a lot of research to link those things together. So that's what we've been working on the last couple of years. And it is really about understanding this relationship. Do healthy soils equal healthy plants, healthy animals, healthy humans? I won't have all the answers today, just a few indications of that, because it's a difficult question, especially when you get to the human. But to that end, we often do on-farm sample collection. We do a lot of work, especially in pasture grazing of livestock, cattle, but also bison. So oftentimes, what we also do, and I'll present you some data about this as well, is that some of our soil health data that I'll present, what we did was we often work with paired farms. So we go to a grass-fed beef farmer and we collect soil samples from the pasture. Oftentimes, we also ask, where's the nearest cornfield? And can you ask your neighbor if we can sample that? Obviously, the cornfield, some of it will be used as livestock feed, although it will go into the commodity market for biofuel. But at least it's a model to study what would happen if we raise animals and finish them out on pasture versus growing feed for them and potentially finishing them in feed yards. And this is some work that I'll present. Alan Franz Luber's soil scientist at USDA collecting some pasture samples. Then oftentimes also we collect TMR samples, we collect pasture samples, and compare those as well for the nutritional composition. So to really start making that link between the soil, plant, and then all the way up to meat and milk samples as well. So the technique that we use sounds fancy. It's not that fancy if we explain it to you, but it's called metabolomics. It's the study of metabolites. And many of these metabolites in soils or plants or animals can serve as nutrients to us or to the plant or to the animal, depending on how far down you go. So we take these samples in these metabolites, which are products of metabolism, whether it be metabolism of soil, plants, or animals. We take these into the lab. We run them through what is called mass spec. It's the same type of testing that is used. If we do drug testing on athletes, for instance, not that we do that, but you use similar equipment for it, it can just detect these small compounds fairly reliably. So that's what we use it for, not to suspend anyone, but to look at what's in our food. Then we typically group these by compounds. I'll talk a lot today about phenols, plant secondary metabolites, better known as antioxidants. For the within the lay crowd. And then we obviously have fatty acids as well that are impacted by grazing practices. And then oftentimes when we overlay these metabolomes, so how does the soil health impact plant quality, nutrient density, and then animal health and nutrient density, which over time I've come to believe are pretty much the same thing. And with metabolomics, we've had this historic focus on protein and fat, while two key nutrients, foods, contain probably thousands to hundreds of thousands of nutrients. Sometimes it's referred to as the dark matter of nutrition. So even if we go on to USDA nutrient reference database, we see 150 foods, and one can convince ourselves that's all that food contains. But there's thousands of hundred thousands of compounds in there. No one really knows how many, but I do know we're scratching the surface. And then oftentimes we overlay this with the human metabolome. So what is the impact here on human health, and how does this impact disease-related gene expression? Getting after the saying, you are what you eat. Now, let's jump into the actual bones of the talk, which is what is the impact of livestock rearing and finishing practices on the nutrient density of meat and milk, and what do the metabolomics findings show? If we look at a high-level overview, one thing I can tell you is that grass-fed isn't grain-fed, and grain-fed isn't grass-fed. You'll say, well, Stephan, of course, that makes sense. We did feel the need to study this, especially looking at a large number of biochemicals. So when we looked at beef and bison, we identified more compounds in beef just because there's more information known about beef. But we looked at 820, or we identified 820 individual biochemicals. And oftentimes when I give these presentations, people say, when I get pushback on this, which is fine, it's important to be challenged. People say there's maybe some differences in omega-3 fatty acids, but that's it. Whether I finish the animals in a feedlot, range for 120 days, or I put them out on pasture. But what we are actually finding, if you look at a broader number of nutrients, not just omega-3 fatty acids, you actually find that you see a 50 to 56% difference in total biochemicals. Some of them upregulated in the grass, some upregulated in the grain. We'll talk about whether that's better or worse. It's not that straightforward always. But what it does imply is that the 50% difference is certainly larger than previously appreciated. They go beyond omega-3 fats, as we'll see later on. And we also are able to look at a broad number of animal health markers. So as much as we obviously raise animals and they can provide nutrition to us, the animal grazes the plants and uptakes these nutrients. First and foremost, it is to improve its own health, and we can potentially benefit from that. We've made similar findings here in bison, which I'll touch on today as well. But really what I want to present on is a project that we started already 2.5 years ago now, two and a half years ago, which we labeled the Beef Nutrient Density Project. So this is in collaboration with the Bionutrient Food Association, who identified a number of farmers and helped with the sample collection. So this is really one of the first large metabolic profiling studies. And we collected beef samples from commercial ranches, 3 steaks each, mostly in North America, but also internationally, because a lot of the work has been historically done in a university setting. But we were particularly interested in what's the current landscape now in grass-fed and grain-fed beef. And can we see certain practices that stand out in terms of improving the nutritional quality? So to that end, we collected soil samples, forage, stool, and meat. And if you look at some of the location farms, as I mentioned, we're heavily concentrated in North America. A few samples out of Australia and the UK, as well as Hawaii. But particularly our focus has been North American farms, and also having a good geographical distribution of those farms. Can we even see if you're in North Dakota, or if you're in Vermont, or if you're in California, can we see patterns that arise when what produces the most nutrient-dense beef, or what we were interested in? So if we look at the fatty acid data first, we'll look at the fatty acid data first, then go into the antioxidants. There's a particular interest in the long-chain omega-3 fatty acids, ALA, EPA, DHA, and DPA. So if I use these abbreviations, these are the very long-chain saturated fatty acids. They're associated with various health benefits in humans. They're anti-inflammatory, they can be antioxidant, they can lower our risk of metabolic diseases such as cancer or heart disease, or at least that's what they're associated with. And then the question becomes can red meat provide a good source of these as well, instead of just consuming fish? That is not to say that one shouldn't consume salmon, but can red meat contribute meaningfully as well? So if we look at the first part here, is the omega-6 to 3 ratio, which is considered a really good biomarker of the amount of omega-6s to omega-3s that are in there. And typically what we see is that the reason why the ratio in grain-fed beef is higher than grass-fed beef is really not because the difference in omega-6s are that large between grass-fed and grain-fed, but it's really the absence of omega-3 fatty acids in grain-fed beef due to the lack of feeding fresh forages, which are rich in this aforementioned compound ALA, which is considered a plant omega-3 fatty acid, even though it is also the dominant fatty acid in meat and in our bodies as well. But lower omega-6 to 3 ratio is typically considered beneficial. What is considered a good ratio? Some suggestions that historical diets have about an omega-6 to 3 ratio of 4-1. Our modern standard American diet provides a ratio of about 18-1, which is all the way up here. So even eating, you could argue eating grain-fed beef can lower this. But what of particular interest to us is, A, what is the average, but also what is the range and what's causing this range? And as you can see, there's a clustering of samples here in grass-fed, and then grain-fed, we see a large variation. And now these ones I question whether they're truly grass-fed. Granted, we source these from the wholesale market, so these were off the shelf, grass-fed, labeled grass-fed. At an omega-6 to 3 ratio of 8 to 10, that's typically feedlot beef. So I don't know, we cannot verify it, but if I were to look at that, I would say questionable whether that is truly grass-fed. Here, on the other hand, we know that these came from feedlots. Some of the levels on top are again off-the-shelf beef, and the lower levels are typically sourced from the smaller, what I sometimes you call artisanal or family farm feedlots, where maybe when you're direct marketing to consumers, some consumers might prefer some of the corn traits to the beef, the marbling, and these are farmers that have lower, feed less grains, shorter finishing phases, maybe it's 30 days on 50% corn, 50% alfalfa hay, just to put some marbling on. And those maintain what we consider at 4 to 5, still considerable grain-fed or omega-6 to 3 fatty acid ratios in grain-fed beef. And I must admit that here, down here, the ones that reach about 2-1, 1-1 are all sourced from farmers that practice what is called what is often called regenerative or rotational grazing practices. So what they have in common is that they typically have very biodiverse pastures. They do not overgraze. They move their animals through the landscape. And the data shows that. And as you can see here, and I guess this was also bound to happen in the study, is that most of the farmers that participated were like, I think we're doing a great job, and can we send in samples to verify that? So that's why we're really bottom heavy here, which is a good thing. And then most of what we got here is not really traceable, unfortunately, because it just came off the shelf. So alpha linoleic acid, forages are rich in alpha linoleic acid, green forages particularly, especially when there's plant diversity. As you can see here, and this is another interest, is just look at the major variation that exists across grass-fed beef. So there was a 10-12-fold variation. Grain-fed, as you can see here, a lack of ALA in the grain-fed beef. And it's just because grains are richer in the omega-6 fatty acid, linoleic acid, and forages are rich in the omega-3 alpha linolenic acid. So you can see that is the case here. And you can, we'll see later on, there's a clear relationship between the content in the forages and the meat, coming back to the saying, cows are what they eat as well. So what this opens up now also is we're doing some work with USDA scientists, and then the particular interest of that is also if can this at some point move towards quality control or authentication of grass-fed beef, especially when combined with a measurement of omega-6 to 3 ratios. So that could be a way of testing it, and this is something we've been working on with farmers as well, especially if you source from a commodity market for large wholesalers. It may come from Australia, Uruguay, and you're not 100% sure what is being fed. This could be a way of authenticating that. So again, here, if we look at the omega-6 linoleic acid, it's hard to imagine that these are truly grass-fed, and these were actually sourced from the wholesale market. And then really the two major omega-3 fatty acids which are considered why grass-fed beef may have additional health benefits or providing omega-3s is really the EPA and the DPA content. Now, one thing that I often think is also underestimated is the saturated fatty acid content. We often hear saturated fat are bad for us. We won't go into a whole discussion on that because we could talk about this for 3 hours and probably not resolve it. But safe to say is that saturated fat isn't saturated fat either. They go all the way from carbon 4 to carbon 24, 26 is the highest you can find in food. But the short end of it is that much like the longer chain polyunsaturated fatty acids, those omega-3 fatty acids that we looked at, you also see the shifts in long-chain saturated fatty acids, which are associated with improvements in metabolic health. So even though the total saturated fat content might not differ, if you can get a shift into compounds such as behanic acid, which is a very long-chain saturated fatty acid, which is actually associated with a decreased risk of metabolic disease, this could be another benefit as well. So you see a more favorable shift in the fatty acid profiles. And the similar findings are made across other species as well. We see here grass-fed bison and grain-fed bison. So here the grass-fed bison had a omega-6 to 3 ratio of about 1.8. Between, like I said, between 1 and 2 is typically what you see when animals are rotationally grazed on species diverse pastures. And what was interesting here, this work we published earlier last year, I should say, we're in 2024 now, but we published this in April of 2023. We worked with the Turner Ranches, which is one of the nation's largest bison producer. And what they do, both grass-fed and grain-fed, but what they've noticed over time is that especially bison don't do that as well in tight confinement. Also, when they're on total mixed rations, they don't do as well. So they move towards a feedlot scenario where they give them free choice access to corn, alfalfa, and then meadow hay, and they also have a lot more space. And what this was really interesting is that the omega-6 to 3 ratio was just above 3 here, which is actually very good for grain fed. And I would be considered if we accept that anything below 4 to 1 is healthy for us. Not to say that something above that is unhealthy. I want to make that clear, but this we accept that this is healthier, at least. That the grain-fed bison, when fed free choice corn and hay, still reached what I would say is a pretty good ratio at 3, which is typical feedlot is about 8 to 10. Another interesting observation is that they, when provided a choice, they ate about 50% corn. So they did their corn, and we're actually following this work up now. Is what if we take it out on pasture now, and we put a feed bunk with some corn or out on pasture? Can we then maybe put on some of the weight without really negatively impacting this omega-6 to 3 ratio? This will be a follow-up study that we do. I must admit, we also are doing a tighter confinement feedlot there, and my guess would be is that even though the current grain-fed bison is considerably lower than feedlot beef, my guess would be if you put bison in tight confinement and feed them a total mixed ration, it will look like more typical grain-fed beef. So there's nothing, I think bison are special, but as far as the metabolism goes, there's nothing special about a bison versus a cow in terms of the omega-6 to 3 ratio. So this is just to kind of note that we were making similar findings across species. So typically grass-fed beef on average, of those are about 350 samples. So over 120 farms or so. And we see here that the grass-fed beef has an omega-6 to 3 ratio on average of 2 to 1. Again, note that incredible variation. So the samples that are in here are also the ones that reach 10 to 1. Grain-fed beef, also a variation between 3 to all the way up to 20, but on average 7. This is from our grass-fed and grain-fed bison data. There's some work on pork as well. Here we can also see if we have corn, soy-free pork plus daily pasture, the omega-6 to 3 ratio reaches about 5. If you give them daily pasture, but give them 50% corn, soy, about 10, and pork that is fed grains without pasture access, it goes all the way up to 30, which is very high. With chicken, it is the same thing. We actually tested, usually when we test the pastured chicken, it is about 8 to 10. So this is what we're seeing here in the middle. This is just corn, soy, fed chicken. When farmers move towards feeding less grains to chickens, and maybe, or I should say not corn and soy, but like peas or oats or barley or some other feed products, we typically see that the omega-6 to 3 ratio drops to about 5. And we did test some chicken also that was supplemented with flax and kelp, which are very rich sources of omega-3s. And they actually reached a ratio of 1 to 1, which initially I did not believe, but it was definitely the case after rerunning the samples, because I could not find this in the literature anywhere, but it is possible even in chicken. And then we have some wild deer samples too as a control. What do wild animals look like? Again, here you can see that the wild deer are about in line with grass-fed animals as well. So you can definitely reach that ratio with good rotational grazing practices where there's no overgrazing, there's plant species diverse pasture. Then the question you always hear, is it a good source or not? There's no official US guidelines on this, but your official European food standards, they consider everything above 40 milligrams per 100 grams of omega-3s as sources of long-chain omega-3. So you could technically make a label claim on that. If we assume that a ribeye is on average about 15 grams of fat, we see that several of these reach that threshold. Again, not to say that it's arbitrary, right? Because if you can get 30 or 20 milligrams of omega-3s from beef, it's still meaningful. We'll see later on. But it is noteworthy, and this is what population-based studies would suggest too in Australians and Irish folks that eat more pasture-based, that it can provide a good source, even though on paper it's not that much if you look at it. And that probably has to do with the bioavailability of this. If I give you a 40 milligram omega-3 supplement, we assume, or we typically see, is that it doesn't raise the levels as high as you would get it from food sources, probably because foods contains those thousands other metabolites that help with the uptake. So food is definitely more powerful than supplements. Is grass-fed beef a good source of omega-3 fatty acids? Despite of what I often read on social media, where people say no, the answer I would argue is yes. Because randomized controlled trials would actually suggest that you can increase the omega-3 profiles in your blood with a few percent when you eat a pound a week of grass-fed versus grain-fed beef, or daily grass-fed beef consumption in another study as well. And there's about now, I think, about 10 to 12 randomized controlled trials, not just on beef, but omega-3 enriched pork or chicken, eggs, and beef that show that you can meaningfully raise the omega-3 profiles. Another one here is in pork, omega-3 enriched pork. We can see here that the DHA content, which is an omega-3 fatty acid, is increased when you consume the omega-3 pork over 12 weeks, whereas there's no statistically significant change in when you eat regular grain-fed pork. Coming back to the fact that omega-3s are considered anti-inflammatory, what this study found is that the inflammation went up when consuming regular pork a bit, and here in this case it did not change. So there is some indication that it may be the case. Now the question is, what impacts this fatty acid profile of meat? So we ran some statistical analysis on that, and we were particularly interested in what we explored a wide variety of things. It was soil health, it was organic matter. Does any of that matter? We didn't find a clear relationship there, at least on an individual basis. The strongest relationship shook out in plant diversity. So more plants on pasture, with it about diminishing return after about 8 to 10 plants, but lower omega-6 to 3 ratios were typically found when there's more plants on pasture. So think of it as kind of if this morning we only had one food to select from, or we had, I don't know how many, 5, 10 different foods to select from, we'd be able to nourish ourselves. A cow is not that different. And this is not to say that it's per se this oasis of 200 plants that's necessary. But what we typically see is that if the field is dominated by a monoculture, that this diminishes the omega-6 to 3 ratio, and it gives a higher ratio. And then another thing that we noticed is, and I will go over it later on, it doesn't even have to be much. It's just those nips on those medicinal plants that really make a difference. Think of it as like throwing some spices on your own food that can make a difference for our health. Same thing for the cow. Now, is there a relationship between soil health, coming back to the cornfield and the biodiverse pasture? This work illustrates we found a relationship with plant number. So this was statistically significant. So better omega-6 to 3 ratio, more plants on pasture. We did not find anything with soil organic matter or other indicators of soil health. We have to do, but we'll see on a group level that's the case, but on an individual level, we could not detect that. Maybe we need more data. I'm not 100% sure. It also tells you that biological systems are incredibly complex. But the plant number and also the percent grazing of the finishing phase made a difference. And that makes sense, of course, if you finish on a pasture, that you see a relationship. Now, the good news is that overall, and this is from a paper we published just a few months ago, soil organic matter appears to be improved on a group level. So remember, we sampled pasture, we sampled cornfields. These were all within a mile, so we could control for any weather conditions or other factors that could impact that. And we typically see if you manage the field as a pasture or you grow commodity feed, then that the pasture, managing it as a pasture, improves soil organic matter, total exchange capacity, as well as various minerals. Now, here's where I think it becomes interesting. Not to say it wasn't interesting before, but this is metabolomics data here. And ergothioneine and histidine betaine, they've been getting more attention in the literature. They're antioxidants, but the nice thing about these compounds is at the present, they're known to only be produced by relatively few organisms, and these are cyanobacteria or fungi within the rhizosphere. So if these appear in your food, there's only one source that they can come from, that's the soil. And what we see here is that healthier soils apparently produces more of these metabolites in the meat. And we have these metabolites in our body too. They're upcycled from the soil to the plant, through the animal. Mushrooms are a very rich source of that because they are, after all, fungi. But this compound has been studied, has been getting interest in human nutrition as well. Then this was the initial antioxidant data. So if we go into these antioxidants, these phytochemicals, these are plant secondary metabolites. We call them secondary in the past because we didn't really know what they did. Primary nutrients would be amino acids and vitamins and minerals, but secondary metabolites, what we now think of as antioxidants. So these are polyphenols, flavonoids, benzoic acids. They give you berries, they're purple color, but they also appear in animal source foods as metabolized versions. So that makes it really interesting as well. If the animal consumes forages or feed that you and I cannot consume and has access to compounds that you and I may not have otherwise access to, is this another way of further increasing the phytochemical richness of diet? Again, here, the story is variation. Grass-fed beef being a little bit of a Wild West. I did not display the 146 because then everything would be kind of shifted. This bar would be very low. But there was a range of 17 to 146 in total antioxidant content. So this is a combination of about 30 antioxidants. On average, 38 in the grass-fed and then about 20.3 in the grain-fed. So about twice as high. But as you can see here, is that the best quote-unquote grain-fed sample is twice as good as the worst grass-fed sample, but the quote-unquote most antioxidant-rich grass-fed sample is 15 times higher, 10 to 15 times higher than the worst grain-fed sample, or still 4 times higher than the best grain-fed sample. So we see this incredible variation. And again, as we'll see later on, the plant diversity is the one that shakes out here. Again, coming back to not to belabor the point, but if we go to a buffet and we have 40 foods to select from, we can better nourish ourselves than when we only have 1 or 2 foods to select from, or being fed a ration. This is true for humans. This is true for cows as well. So plant phenolic compounds, these are major parent compounds: quinate, benzoate, cinnamic acid. Cinnamic acid is found in nearly every plant, but it's very rich in cinnamon, but you find it in other plants too. So that's what it's named after. Oftentimes these things are named after the food that they're found in. Phenolic acid, also rich in almost every plant, but also rich in vanilla. Prunin, being rich in prunes, but also rich in other plant foods. So we're not that original in science with naming things, but that's okay. But we know these plant phenolic compounds, and this has always been the tricky part. You study this in a plant, but this is not how it appears in animal source foods. It appears as hydroxylated versions. It appears as hyporate, which is produced from phenolic compounds. So this has always been a difficult thing to figure out, and we're still learning a lot about these pathways and what we exactly need to measure, because if we measure this and this, we don't really detect much. But if we measure this and this, then the story starts to appear. So this is grass over grain. Again, here, twice as high, 3 times as high. 4-ethylphenyl sulfate is 20 times higher. And this is also further downstream of this. So maybe there's a case that this gets metabolized and metabolized. Then the interesting question becomes, and I do not have the answer for this today, maybe in a few years, is we know that, well, stachyderine is very rich in alfalfa, and then we don't typically consume alfalfa. But if we have these metabolized versions of that normally has to be processed by our gut microbes, and I'll stay here so I don't walk away from the microphone, even though I could take a little walk, is that what if we consume these pre-metabolized versions? Because normally they'd be metabolized in our gut microbes, they'll be metabolized in our liver, but now we are providing a direct source of that pre-metabolized. Does that matter? I don't know, but it could. It's been something that's been on our mind, at least, if there's a benefit of getting some of these compounds, these metabolized versions. So we get quinate and benzoate if we eat the plant, but we get these when we eat the animal. Piperine and phagoamine, they're rich in wheat and grains. Not to say that, like I said, it's never black and white. These are antioxidants too, and they're higher in grain fat. So here, quinate, benzoate, high in plants, and then hyporate when it gets metabolized. And as you can see here, in the previous data, we looked at, this was untargeted metabolomics. So we were looking at what the hell is even in here. And on this work, we try to quantify it as well, which we did. And again, find that grass-fed beef is higher than grain-fed beef. And this is an antioxidant. It has received some good amount of work in humans. and associated with reduced odds of metabolic syndrome. And it's a biomarker of phenolic intakes in mammals, because the phenol ring of these compounds gets combined with hyporic, becoming hyporic acid. Chlorogenic acid, same story. Again, here, this variation starts to appear, also considered higher. Chlorogenic acid downstream of cinnamic acid. Again, here, these are all antioxidants. All phytochemicals are antioxidants. They just work on slightly different pathways. Now, you may ask, Stephan, how can you be so sure that there's a transition from forage to animal? Because we measure benzoate in the forage, in the grass, and we measure benzoate in the corn-based TMR. And then we measure hyporate. Benzoate becomes hyporate. High in the forage, again, this variation, higher in grass-fed. It mirrors each other. Coming back to what my grandma told me, Stephan, you are what you eat. And this is true for a cow as well. This is the clearest way to say it. Cows are what they eat. If we look at total plant secondary metabolites or phytochemicals, we see these are forage samples. So this is the pasture samples. We see here, again, variation, but it becomes clear that there's about 4,000 units in the forage samples. There were a few hundred units in the corn-based TMR. And we were also particularly interested in what is actually coming from the corn versus what is coming from the hay. And it turns out not much is coming from the corn. So while this can be a good source of protein and energy, it does not provide many antioxidants, almost none, actually. So the corn-based TMR is what really is providing that. And then if we look at vitamin E, which is also a phytochemical, an antioxidant, but we know that is an essential nutrient, so we label it a primary nutrient. But grass-fed here, statistically significantly higher, about twice as high. And then if we look at the forage, it is about 20 times higher than TMR. So the question we had here is that there's a contribution from the pasture phase, we think here, in the grain fed, because it seems to drop off a lot in their diet during the finishing phase. And this being fat soluble, you just see this, it probably starts here, and then you see this drop off as the animal goes into the feedlot, because the TMR is not really providing this. So a little contribution from TMR. And the golden rule in science is, if you could think about it, someone else has thought about it before you, and that being the case, phytochemicals. This is work in 1987 already, done in NC State. Days on TMR, you see here the phytochemicals parts per million started at 120. After 60 days, they dropped to about 50. And then you see a further decrease to about 30. So 3 to 4 times reduction here. And an Italian group under the leadership of Priolo, who's done a lot of work, especially in milk, finds the same thing in lamb. And it happens in bison too. So here, bison, same compounds measured in bison. Hyporate, 3 times as high, 4-alpha-phenol sulfate, 18 times as high. But then stachydrine here, alfalfa compound, higher, because these bison in the feedlot were consuming 50% alfalfa. And then I'll read this to you, but the interesting part is that, and you'll probably say, for those of you who are grazing pastures, and Stephan, yes, I already knew this because I observed this day in, day out. And that might indeed be the case. And if you look at older writing, this was already known. Clearly, because Warren Angus Ferris, who was an explorer in the 1800s, he traveled the Great Plains from 1830 to 1835, and they fed on bison. And then in his book, he writes something amongst the lines of, when they ate the bison in poor flesh were the worst diet imaginable, but as they fed on diverse mixtures of plants, no other meat could compare. And he writes, with it, we require no seasoning. We boil, roast, or fry it as we please and live upon it solely. And what seems most singular, we never tired or disrelish it, which would be the case with almost any other meat. Now, what I'm pretty sure he's describing is the phytochemical richness of that meat, because I talked about it in terms of health context, but these are things that you can taste as well. So it's kind of like marinating spices from the inside out. So I am fairly certain that is what he's referring to, fast forward 200 years, that more plant species diversity makes for and I know taste is subjective, but it makes for probably at least a biochemically more complex piece of meat, and something that apparently Warren Angus Ferris could pick up on in bison as they roamed through the Great Plains. So are flavor and health related is a big question. Now, let's finish up with animal health. As I mentioned, my background is in muscle biology. So we used to study, and we haven't done it in a while, but studied muscle of humans. So we take muscle out of a leg. It's a small piece of muscle. It sounds the worst that it is, but we think about a pea size. I had 8 of these and I'm still walking. But we take a pea size piece of muscle out of a human's leg with a biopsy, usually the thigh. And we did this in athletes, like runners, cyclists, and we did this in people with metabolic syndrome. Now, when we started doing this on an animal, not to say that animals in feedlots get metabolic syndrome. I don't want to imply that is the case, but you do see sort of these parallels amongst us as mammals, or if you study in lab rats, right? Very different diets, different phenotypes within the muscle. So this is studied in the muscle, oxidative stress. Here, what we see here, if you have higher levels of under oxidative stress, we see that allantoin goes up, urate is converted to allantoin. So it goes up here in the grain-fed bison and is lower here in the grass-fed bison, and urate remains higher. So we consider this to be less oxidative stress on this biomarker. Then another major biomarker that is of interest is called 4-HNE. If you Google 4-HNE and cancer, you'll be reading for the next couple of years because it's been studied so much. It is also one of the reasons why the International Agency Research on Cancer from the World Health Organization labels red meat a potential carcinogen. Again, it'll be a very long discussion, but the short end of it is that if you go to page 167 of the report, that it is processed meat, actually, that's causing this association. So, but safe to say is that 4-HNE, it's a product of lipid peroxidation, and this was apparently very interesting to us, is that we know if we have more oxidative stress, we have more 4-HNE. Apparently on a cow, it's not that different. So how you handle the cows, also the cattle, makes an impact on 4-HNE, and I don't know the answer. What does that then do to us? I'm not sure, but there's some concern about dietary sources of 4-HNE. And if you can lower this by how the animal is being raised, even you can see this here in the grain fed, again, here coming back to the artisanal feedlots, seeing lower values. Again, here, they're not black and white for sure, but note the individual variation. But on a group level, we do consistently see that grass-fed has a healthier phenotype. And again, here, coming back to the fact where we study this in humans, we take muscle out of an endurance trained athlete, a runner. They have more mitochondrial metabolites. Mitochondria are the life of our cells, the powerhouse of our cells, produce a lot of energy. If you can produce this through mitochondria, it's typically a good thing. And all this shows is that it's a more athletic phenotype in the grass-fed animals. Again, now with the bison, we're trying to figure out, because we can do controlled studies on those with the Turner ranches. What if we restrict their movement on pasture? Or what if we increase their movement in the feedlot? So what is the impact on these things from feed versus movement? This is a major question, because we also know that omega-3 fatty acids are preferentially metabolized by the mitochondria. So having more omega-3 fatty acids is also driving this metabolism. So we think it's a contribution of both, but we don't know what it is. But it is really interesting. Again, I don't want to imply here that these animals are per se unhealthy, but we do show a more athletic phenotype here in the grass-fed animals. And final thing also is the gut microbiome health. Again, here, bile acids with anti-inflammatory effects higher in the grass-fed animals. They could have an improved gut microbiome as well due to what we think, due to the plant diversity. So this is the work that we published in the bison, and really, for us, this was quite eye-opening as well. And it convinced me that animal health and nutrient density, it's essentially the same thing. They're very much intertwined, and it's an arbitrary cutoff. Because the first half of the presentation that I gave about nutrient density, if I took that piece of muscle out of a human and I'd give the same presentation, say, oh, this human is very nutrient dense, everyone would be looking at me like you are crazy, right? And hopefully you didn't look at me when I did that when presenting about cattle, but it's the same thing. So here, the animal health markers, the glucose metabolic health was a little bit decreased in the pan-finished animals. But again, here, this is not a true feedlot. This is a feedlot spa for bison. Oxidative stress was still higher than the pasture-finished animals, but the pasture-finished animals, better mitochondria, more phenolics, more of vitamin A and E, more long-chain saturated fatty acids, omega-3 fatty acids, and less of this compound 4-HNE. But then on the other hand, more vitamin B5 and 6 in the pan-finished animals. Grains are rich in this. Gamma-tocopherol and antioxidant rich in grains too. So certainly, I want to make sure I don't paint with broad strokes, is that there's definitely a nuance to that. And coming back to the interest of what if we provide some grains or some byproducts, I think the intention of feeding byproducts or grains to an animal could be a good thing, right? It's just when you take this to 60, 70% or so, maybe then it becomes a little bit of an issue. But in and of itself, it may not be a bad thing to provide some grains to animals. And we certainly have some samples that I guess we labeled grain fed, but they were on grass, and then last 30 days they went on corn for a little bit. So Those are interesting things to look at. And there's some other work also with, not our work, but another study that did grain finishing or fed some grain on supplemental grain on pasture and actually found some benefits there as well in terms of putting on weight without having many negative effects on the animal's health or the omega-3 fatty acids. So there's a balance to be gained potentially there. Now, connection to human health, this is where it becomes murky. I think my laser pointer stopped, but we'll see the computer still. But the connection to human health, yes, it does. Okay, perfect. So the health effects of pastured meat consumption, there's one study that always comes up, but it also tells you the state of the research right now. Forage fat and grain fat, this is kangaroo, study done in Australia. Wagyu beef versus kangaroo. 2 hours after the meal, the forage fat animal, which is the kangaroo. Inflammation does, yeah, I see a little bit of an inflammatory response after eating. This is normal, this is important. But if it becomes excessive and becomes chronic systemic inflammation because you have a meal after meal is when people run in trouble, we see that the forage fat as a reduced response, which can be considered a good thing, but this is kangaroo. So we just replicated the study. I started at Utah State 2 years ago, and I think last time when I presented, I was saying we're funding for it and trying to do the study. We did the study, but we finished participant 34 almost. One more person has to come back to eat an Impossible Burger because that was the third arm, the Impossible Burger. So I don't have the data now, but we've replicated the study with grass-fed and grain-fed beef. We'll see what comes up. But the conclusions and directions is there's a 50-60% difference in abundance of compounds. Phytonutrients become especially concentrated in the meat and milk, and we see that there's where a lot of the difference are, the secondary metabolites that were previously underestimated. It's not the amino acids that make a difference. You have the same amount of essential amino acids, grass-fed and grain-fed, but it's really in the omega-3 fatty acids and these phytochemicals, the carotenoids, vitamin A, vitamin E, these antioxidants where you see differences. Better saturated fatty acid profile, and then less of maybe stuff we don't want, 4-H&E and homocysteine. The conclusion maybe we have is that what we do to our animals, we may do to ourselves, and a more healthier animal, more nutrient-dense food. And I think here's where obviously the question is, well, Stephan, you can eat these phytonutrients just from eating plants. To an extent you can, yes. And I won't say that a carrot is a better source of beta-carotene than grass-fed beef, even though beta-carotene is 3 times as high in grass-fed beef versus grain-fed beef. It's still a lot lower than in a carrot. But I think here's where it becomes unique, is that herbivores, they consume plant species that we otherwise cannot consume, and they also metabolize these compounds. And now the question is, what does this do to us? And unfortunately, I don't have the answer for you today, but we will measure some of these things in humans now to see if we get more 4-hydroxybenzoic acid or hyporic acid in our body, does that make a difference for us or not? I do want to also note that our data does not per se indicate that grain-fed meat is therefore unhealthy. We've recently completed a study where we fed people all foods from regenerative agriculture versus conventional agriculture. The regenerative agriculture foods were all fruits, vegetables, unprocessed meat. We matched those by just store-bought non-organic produce, but also meat, milk, and eggs. But it's 90% whole foods. And what we see then, if the people that we recruited people that were in standard American diets, everyone gets healthier. So I'd say, just to say it plain simple, if you look at the fries, a Coke, and a burger, if you're going to switch out grass-fed beef for grain-fed beef there, or vice versa, this is not what moves the lever on your health, is what I want to say. So we see that people eating grain-fed beef, if they do it as part of a very healthy diet, their health improves compared to consuming a standard American diet rich in ultra-processed food. So that's, I think, an important elephant in the room. Not to say that those details won't matter over time, right? But you probably get 80% of the way there by not eating a bunch of ultra-processed foods, I think. So we compared grass-fed beef, grain-fed beef, and plant-based meat alternatives. We completed this over the last year, this study. We ran 34 participants, so it's a big study. So this year, we're set to do the analysis on that to try to replicate the kangaroo study, essentially. After eating grass-fed beef, grain-fed beef, or an Impossible Burger, because I left the workout today, but an Impossible Burger is not grass-fed beef or grain-fed beef. It's very different, 90% different in that case, with using the same metabolomics analysis. So even though the protein might be matched or the fat content might be matched, you're studying a plant and an animal, day and night difference. Not to say one is healthier, but they're just completely different food sources. And I think that's important to note. So with that, lots of more questions that came up. But I think we can conclude that there are some additional health-promoting phytochemicals that become concentrated in the meat and milk on animals when they're finished on biodiverse pastures. So potentially additional health-promoting compounds that are being accumulated. And I think their presence has remained largely underappreciated up until this point. But it's just newer techniques enable better work. And with that, this is simply the messenger. There's a lot of people that worked on this, various folks from Utah State University, a whole list. Camille is here today as well, who worked on this. Folks at Duke, where we really started this work in my last year at Duke. And then folks at USDA ARS, the Bionutrient Institute. And we did the bison work in collaboration with South Dakota State. And then various funders as well of the work, a combination between USDA funds and industry groups, as well as some philanthropical funds. So with that, thank you so much. And I think we have about 15, 20 minutes for questions. Perfect. All right. Thank you so much.
Okay, so questions from the fellow in the front row.
So I have a question about, so your research is based on pasture grasslands. My question is, what's the pasture management? Is it all over the board? Are you using it as a marker? So if you got good pasture management with the rest periods and everything else, or just people are just throwing them out? Because where I live, it's all over the board. I couldn't conclude anything from what you just said, because there's people that overgraze, there's people that just throw them out. There's so variable in the pasture forage part. How do you quantify any of that?
It is questionnaires. At least in the big beef nutrient density project, we ask people questionnaires, but as you see, we were very shifted towards the rotational grazing. I think 70% of the grass-fed respondents said, or 80% said that we rotationally graze. So moving the animals out to the pasture. But it is true indeed that with continuous grazing, we've had a few of those, and those do seem to sink a little bit towards the bottom. But yeah, it is a good question on that.
I can tell you, I'm regenerative and I can self-anoint myself as whatever. Like the questionnaire doesn't, until you put feet on the ground and quantify, right? That's the details. The devil's in the details.
Yeah, that's right. No, it's a good point, and that's additional work that certainly needs to be done. Might have to take a sabbatical for a year and visit all the farms, which would actually be good. But yeah, that would help. This is a way of kind of opening up the door, of course, to understand what the differences are. But yeah, observationally, I'd say, I would hypothesize that the continuous grazing and overgrazing is, and we only have a few farms that we can glean this from, does limit the nutrient density. And there's some interventional work to suggest with continuous grazing is that you have continuous grazing, it stimulates more of a monoculture, you have more omega-6s in that. So it's an excellent point. And I think that's also why we find the data to be a wild west in grass-fed. Yeah, I appreciate that point.
So related to that, I just wanted to jump on Troy's question. We had a question online related to this about seasonal variation and changes. And so does your surveying now take into account any seasonal variation?
At the moment, not much, because we actually wanted to get everything from the end of the grazing season as much as possible so that we could see it independent of seasonal variation and also independent of geographical location. And what the data would suggest is that if you're in North Dakota or North Carolina, if you graze using locally within a local context, there's nothing special about being in North Dakota versus North Carolina if you're doing a good job. And of course, it's like you can have a bigger discussion on this as well, whether raising Black Angus in the desert of Arizona makes sense, but that's a deeper discussion, of course, right? And maybe it's better to raise some other animals there. But yeah, that's typically what we find. But yeah, the seasonal variation, again, here in this work, we did not look at that, but there's been some interventional work, some nice interventional work, especially done from the French equivalent of the USDA. And they find this sort of seasonal variation that if you come out of the winter, it goes up over the grazing season. It stays high, sometimes plateaus, depending on what your summer is. And then it comes down again. So you do see the seasonal variation during the grazing season being more complex, the beef.
Thank you.
Great, Stephan, thanks for your work. I'm very impressed. I had a curiosity about visual appraisal. So do you include in your testing and looking at the steak? Years ago, we sent samples to Susan Duckett at Clemson, and she precipitated all the fat out and gave us a grade equivalent to USDA grade. And she could see that we fed just grass because of 1.2 to 1 ratio. But when I asked her the question is, can you see the fat like a USDA grader? She said, you can't see the omega-3 fats as well as you can see the omega-6 fats. And then you realize the whole grading system, the whole ultrasound programs are developed on corn-fed visual appraisal. So it's a big question in the industry, particularly USDA just Friday rolled out a pilot program for grading in small plants where they'll just take a picture. And then you go to that, the visibility of the fats. Do you have any thoughts or comments on that?
Yeah, I don't have the expertise in that area, so we did not systematically look at, but I'm also not blind. So I can see, of course, that when we process the meat, yeah, if animals are on grass, it typically has a more orange tint to it. So the carotenoids, and that's what shakes out in the data. So that's what you're seeing, this orange tint of the carotenoids. And again, here, this is just, again, visually, we did test some meat from old dairy cows, a few of them, and yeah, those were really orange because they're fat soluble. So over time, years of accumulation of carotenoids from the forages into the meat gives a sort of this deep orange color. So that is something you can see. But yeah, it is a good point with the grading system that if measures on like the thickness of fat and things such as that, is that you're stacking it maybe in the favor of grain fat potentially. But yeah.
I just have a question for you. You briefly touched on it, but how does stored feeds compare to pasture? And what about baleage or fermented forages compared to pasture?
They're about intermediate. So they fall in about intermediate between, I don't have a slide today, but they fall about intermediate. So if you take the pasture and you turn it into baleage, or that it becomes lower, you see a reduction in these phytochemicals, but you stay higher than TMR. So you kind of fall in the middle of it in terms of that. But again, an important point there is that you see this also the seasonal variation, but is that if you use a biodiverse pasture or forage, that could help maintain some of these phytochemicals. But the nature is that if you want whole year round fresh beef supply, is that this is a consideration, or milk, something. A gentleman in front of me has a milk on his shirt. But this is something, you notice very much in milk as well. And if you ask me, the milk in the spring that I get from the local farmer tastes better than in the winter, but I still like drinking it in the winter.
Okay, just to add an element of excitement into the room, you get a book, and it is Grass-Fed Beef for a Post-Pandemic World, written by Red Shin. So we're just randomly going to give prizes to people who ask questions. All right.
So maybe it's just because I've been doing this too long, but I was at a conference in, I don't know where it was. Dr. Diamond out of Utah told us this 25 years ago, that grass-fed butter is not food, it's medicine. So you're just putting a whole lot more information to what we talked about 25 years ago. And this is the part that makes old grumpy farmers now, 25 years later, we're have to battle against fake meat. What are we doing with this good information? Farmers know this?
Yeah.
How is the information getting out to the people that need to know it?
Yeah. And no, it's an excellent question. I don't know what your feelings are about it, but I do feel like even though the market share is smaller, the work would suggest it is on the rise for sure. And the direct to consumer marketing is probably more now so today than in the past. But if you look at it overall, it makes up a small market. And oftentimes when I talk to wholesalers, then I'm like, why are you sourcing all of this from Australia and Uruguay when we have a really good grass-fed beef supply here? And then usually it is, if I want to, how many tons is, I have to source this from 150 ones. So I think it's sort of these things, supply chain issue, ability to process. And I think that's getting better, my understanding. But yeah, there are supply chain constraints. Now with social media, there's some opportunities. So that's I think it would command a premium is if you're able to direct market. But I agree, for this to move the needle, it needs to be the Costco's of this world and the Walmart's of this world that start to pull this as well.
Just a brief response. Someone recommended an organization called Good Meat. I think that's helps you promote. Okay. My question was, and maybe I missed it in your presentation, but were the pasture diversity samples taken from the same farms that you were sampling the grass-fed beef from?
Yeah. And
So that's somewhat of an indicator of the health. Was that pasture diversity?
Yeah,
The
that's
health
right.
of the system?
Yep. No, it's an excellent point. Yeah, it was. And a lot of these, what I presented on there was the work that was from where we actually went to also to the farmers. And you do see this, you definitely see it shaking out with the forage quality and then the chemicals that you see in the meat. And on that, actually, speaking of seasonal variation, is we saw the seasonal variation in the forages because we went to those farms 3 times over the grazing season. This is 2 years ago. And that it peaked in around May, June, still pretty high at the end of the summer, and then went down in October, November, speaking to that seasonal variation. It's an initial indicator. And like I said, there's like bits and pieces in there, like soil health seems to be improved if you manage it, but finding this direct relationship in inherently messy biological systems is difficult. Yeah.
Yes, thanks for your presentation. It was great. So I have a 2-part question here. So in your research, let's say you're doing research on the bioavailability of the meat and the nutrients. Do you see a relationship between the anti-nutrients? So if you were to eat that meat with a bunch of plants, do you see a reduction in the bioavailability of the meat? And then the other part of the question is, has this research changed how you and your family eat and what you consume? Have you changed your diet due to this research?
Yeah, the first question I don't know the answer to yet, so I'm not sure how this would impact the anti-nutrients. What I can say broadly, is that these compounds such as 4-H&E, when you eat meat with spices, you reduce these compounds quite a bit already, or you marinate the meat, or you eat it with vegetables. So you see a beneficial effect on that as well. But specifically in the context of grass-fed beef, I don't know the answer at the moment. And then the second question is, I'd say yes, I'm human too. As much as we try to be objective as scientists, I have biases. And if I look at this data day in, day out, then yeah, I buy a lot of local pastured meat, local milk as well. So yeah, this obviously, and I grew up with that because that's obviously what my interest was. I grew up close to farming systems. We get local milk directly from the dairy. And as I got in college, I maybe went off the deep end a little bit with diet, and that's why you go to college. But then after that, yes, as we got more into this work, for sure. But then again, I also learned that I'm also not afraid to eat some feedlot beef or something like that, right? If my diet quality is incredibly high otherwise, and eating a bunch of unprocessed fruits and vegetables, then yeah, I think we're really also at that point also studying details at some point. So I think my point being is that because I know grass-fed beef is more expensive, even if in the socioeconomic status that it may be not affordable, I don't think it's unhealthy to eat feedlot finished beef or conventional meat if you leave the coke and fries away. So.
Yeah, thank you. Great presentation, very rich. And I think this question is actually somewhat of a follow-up to that other question. I assume that all of the testing was done with raw meat. What is the likelihood that any of these chemicals that we're looking at degrade during the cooking process?
There's a likelihood of that. Yes, some of it will degrade during the cooking process with heat. So the bison was cooked in that study. The beef was not. So we still find surface magnitude there, but there's some work done. This is not our work, but again, coming back to the French and Italians that have done this work historically in cheese. What they found was if they pasteurize the cheeses, that there's a reduction of 30 to 50% in these compounds as opposed to raw milk cheese. I'd say the French and Italians, right? It's sometimes a little bit considered blasphemy to pasteurize your milk. And they're doing this presumably for the flavor compounds of that are retained. So they think of these phytochemicals. As a human nutrition scientist, I think of these as health promoting, but they're also flavor metabolites. And I think that's what Mark Schatzker today will talk about later on, was the flavor of these things. But yes, they reduce them. And I think why the French and Italians don't do it historically is because they feel like they're sacrificing flavor on this. And this is clear from the papers that the Italians published in the early 2000s as well, where they talk about these as flavor rather than health.
It may not have been a focus of your study, but what can you say about the pass-through of these chemicals to the next generation of the animal? Particularly interested in poultry, but I guess it would apply to other animal groups as well.
Yeah, it's an excellent point. I just sent in a grant a few weeks ago on that. So where we were looking at, and I think we'll try to continue that work. There's a colleague, Juan Villalba at USU, who's looking at more health, methane, pollinators over with biodiverse pasture, but then also following it through the calf. So we're going to presumably collect blood from the mother and the calf. We'll look at the milk, and then we follow the calf all the way through until slaughter to see if there's epigenetic effects. There is some older work to suggest from one of collaborators, Dr. Fred Provenza, that would suggest that in utero experience to these chemicals can be beneficial and actually can train the animal to eat the or capitalize on the plants within a local ecosystem. So there is a likelihood that this gets transferred in utero already, and the in utero experience can shape it. And if work in rodents, just another mammal, is any indicator, we should see epigenetic changes on that as well. And we notice from humans, we notice from rodents. I see no reason why it wouldn't happen in a cow.
I have a question. Oh, sorry.
All right. Fine. Sorry, Nico.
No problem.
It seems like you have your hands full already with all these good phytochemicals and beneficial chemicals. Do you have any plans to study harmful chemicals like pesticide buildup or like PFAS or anything?
I won't do it in our lab just because it will be too much to do. But I'm talking to colleagues at USDA ARS that's specialized in PFAS, and they are interested, especially since we have samples from all across the nation. And they know what there are certain PFAS hotspots, and they have looked at meat and milk in the past and found that if it's close to a plant that produces products that contain PFAS, that they can see this in higher levels in the meat and milk. So there are some talks on, we'll see if it materializes, but we are, this could be a future area of research for sure to see what's the PFAS content and other potentially harmful chemicals. But that will be through a collaboration with hopefully USDRS.
Yes, about 4 or 5 years ago, I spent a good week trying to find a lab in Canada and the US that does this kind of testing commercially. I was unsuccessful. Is there something out there other than university labs that can do a complex and comprehensive nutrient analysis that I could send my personal beef to and then compare to a data set that you and other university researchers have as a benchmark and say, hey, I'm doing really good, or I can improve this, or I might need more diversity or whatever, or have even a marketing claim for my own place saying, look, we tested our stuff, we know it's better.
Yeah, it's a good question. They're larger companies, but I think the focus is not so much on the secondary metabolites, but they might be able to run fatty acid panels on them and look at minerals and maybe a vitamin content. We're super busy, but we can talk afterwards if you've waited 4 years and you can wait another 6 months potentially, then we are happy to talk about helping you. But yeah, I'd say this, especially the research on the metabolomics, the antioxidants, it's such the early stages that I think we have to, I guess they what I call our research, we have to redo it a couple of times sometimes to figure out how to do it best. So I think it will come further over time as this market grows. But yeah, I mean, we'd be happy to potentially help with some of the analysis. Yeah.
So I'm going to cut it off there only for time, not for lack of questions, because I think we could probably go on for quite a bit longer. And Stephan, you're here throughout all of today. So if folks have any questions at all, or ask them online as well through Huva, we'll get you answers if we can possibly do that. So thank you again, Stephan. Yeah. So we're going to take a break to the bottom of the hour. When you come back, I'm going to ask that you reshuffle a little bit to fill in the holes. So we've got some very full tables, but we have tables with seating holes at them, spaces at them. You won't need to look up at the screen, so it's table work. So I'm going to encourage you to sit with somebody new if you possibly can. Split up couples if you possibly can, or groups of people that work together. I encourage us, mix it up. And folks who said that they would help me facilitate, please meet me over there. So see you at the bottom of the hour.